Hi everyone. So today I want to talk about using the chi-squared test for a genome-wide association study. So this is the Pearson chi-squared test. So the chi-squared test again is with this test statistic here. Okay. So usually we have these genome-wide association studies, or we call them GWAS. So you can think of this as like Inspector Gadget, kind of scanning the whole genome. So what is a genome-wide association study? So these studies involve scanning markers across the genomes of many people to find genetic variations that are associated with a particular disease. So which individuals, have a phenotype in a population, and of those individuals, what kinds of genes do they have that could be associated with the disease in general? That's what we're trying to look for. So pretty much in a genome-wide association study, we rapidly scan markers across the complete set of DNA or genomes of many people to find genetic variations associated with a particular disease. Once new genetic associations are identified, researchers can use the information to develop better strategies to detect, treat, and prevent the disease. So we can find, what kind of genetic variants, what genes along the loci of different chromosomes could be associated with the different phenotypes that we are observing. So that's what we try to do with GWAS studies. So look within the chromosome information, try to find markers of genetic markers of the disease that we're interested in. So in this case, let's just suppose we are interested in looking at type 1 diabetes. So type 1 diabetes, also known as juvenile diabetes, is a chronic condition in which the pancreas, our pancreas is producing little to no insulin. So it typically appears in adolescence, and symptoms can include thirst, frequent urination and going to the bathroom a lot, hunger, fatigue, and blurred vision. So the treatment usually involves maintaining normal blood sugar levels through regular monitoring, insulin therapy, diet, and exercise. So here, for instance, this is a photo of my grandma actually had diabetes, my late grandma. So this is me when I was a little, child, little pipsqueak, and that's my younger brother and my older sister, and that's my maternal grandma. I really miss her a lot, but she suffered from diabetes. So this is a pretty personal disease to me. So genome-wide association studies have looked in type 2 diabetes, for instance, that tried to find risk variants that could be associated with type 2 diabetes. And you can see here that recently they've been looking in type 1 diabetes, tried to discover associations right now that are currently weak. This is about like a few years back, and they're looking at different risk loci that could be associated with type 1 diabetes. So what we find from these genome-wide association studies are these single nucleotide polymorphisms. So what are they? They're pretty much the most common type of genetic variant. So kind of imagine, Buzz Lightyear telling Woody, single nucleotide polymorphism. They're everywhere. So it's basically a change of one DNA base in one location of the genome. Like see here, for instance, we can see an A, a G, and a T. And they are basically one spot along the genome. A lot of people have variation in that spot. Pretty much almost like 99% of our DNA with other humans is almost pretty much identical. But they're kind of like a few 100,000 DNA bases of ours that can differ. But what's important is that SNPs usually are they're really frequently different between certain groups in the population. And it's just one change of a DNA base at a specific spot in the genome. So you can imagine that she has a G in that location, whereas this other person has a C in that location as well. So just a change of one DNA base in a location of the genome. And that could lead to different alleles of the same gene. So let's suppose that we want to look at type 1 diabetes, this juvenile diabetes, And we're looking at the population A, so it's some subset here in the population around the world that we're interested in, maybe just some group or something, you know, we're trying to measure it. So we're trying to figure out, okay, well, we have calculated the genotype expression of individuals in population A at a particular SNP, the single nucleotide polymorphism. So we found that, you know, we've calculated it. We found, you know, 28 people have CC here and they have the disease. 95 people have CC, but they have do not have the disease, so they're controls. So basically the most important thing is the first part is it's like a case control. So this is the case, the people with the type 1 diabetes, the disease. And here is the control. These are the individuals who do not have the disease. And we found three main genotypes. Individuals have a C and a C, so one from mom and one from dad, or a C and a G, or a G and a G. And this is the breakdown of the different you know, genotypes for those individuals, and these are the phenotypes over here. So pretty much like the way that you can think about this is the following, where let me try to write this. Well, here, this is the pheno, and at the top is the genome. And because it's case control, so this is the total number of people with CC allele. So we could think of like when you have a chromosome and we're looking at a specific spot along this chromosome, and then we have one from the same chromosome, sorry, I'm like bad at drawing, mom, and then we have it from dad. And then we're looking here in this specific spot here for the mom. And then let's say that in the dad, it could if it's the same, it would be the C and the C, for instance. Or maybe it could be different. Maybe it could just be like it could be orange instead for the dad. In this case, it would be like a C and a G. This would be a G then. So we're trying to, or it could be a G and a G. Maybe this is also like, mom's also a G, for instance. And the same is true the other way around as well, where, you know, the like either or is possible, either combination is possible. So what we're trying to do is we have this case, and then we have this control information and this phenotype and genotype information for these individuals. So again, this is what I was sort of mentioning, that we have CC, and then we find these the SNP here. This guy's analyzing what are the common genotypes that we found. So we can have a CC, so both C alleles from parents. We can have a CG, which means like one C from one parent, could be mom or dad, and then one G from the other parent. And a GG, so both G alleles from parents. So these are the different like genotypes that we observe and the associated phenotypes. So if we take 28 plus 95, that would be 123. So we have 123 individuals with CC, and then we observe this breakdown of individuals with the disease and the control. So we can try to say, is there some sort of connection between this genotype and the phenotype? Like, do any of these three groups seem likelier to have the disease than the other group? So that's the main question we're focusing on. So is there an association between the genotype of the single nucleotide polymorphism at this location in the chromosome? And remember, we always have two of them because we one from each parent. So we could look at CC, CG, and or GG. If we look at the genotype and the disease status within the population, does having one genotype make you likely to have the disease, type 1 diabetes in this case, than the other? That's what we're trying to say. Is there some association? Is there some sort of a connection that we will then have to investigate further? That's what we're trying to do. Like, so the main question again is there an association between the genotype of this SNP, and here we have 3 genotypes, and the disease status within the population A? So there are many different methods to test this association, and I'll be talking about 2 of them, but in this video, I'll be focused on the Pearson's chi-squared test. That's what we're going to focus on over here. So please stay tuned for the next video, and please subscribe to my channel again and like them if you find them useful, these videos. Thank you guys, and ask me any questions if you have any. So in the Pearson's chi-square test to determine if there is an association between the genotype of this SNP and the disease status within the population A. So our null hypothesis says there is no association between the genotype of this SNP and the disease status within population A. So that is our belief. So we currently believe that it doesn't really matter what our genotype is, if it's a CC, a CG, or a GG, it doesn't really matter. It doesn't have any association with having type 1 diabetes. We're not going to find individuals who are likelier to have it based on the genotype of that SNP. That SNP is not important. That's what our null hypothesis is saying. So in other ways, what it's trying to say is that we just believe that the SNP, so it's saying that the SNP is not important. That location along the genome is not important. But the alternative hypothesis is saying there is an association between the genotype of the SNP and the disease status within population A. That could be a protective effect, that genotype can protect us from the disease, or maybe it leads to it, but the SNP is important. That's what the alternative is saying. So the smiley is very shocked. It's like it's an important SNP. So basically, guys, I can a chi-square test, according to Wikipedia, is it's also written as a chi-square test. It's a statistical hypothesis test that's valid to perform when the test statistic is a chi-square distribution under the null, and it's trying to determine if there's a statistically significant difference between the expected values and the observed values, like the frequencies, in one or more categories of a contingency table. So you can think of a contingency table as, like, the case and control table that we had for right now for the type 1 diabetes, where we have like the genotype and then information on like the phenotype, cases or controls, and then the columns were like the different genotypes as well. And we're trying to observe if there is a statistically significant difference between what we expect and what is observed. And this is what we try to do with a chi-square test, using a contingency table. And we're going to go into that some more. So just stay tuned right now. So if you think of a chi-square test, so imagine we have a city with 1 million residents with 4 neighborhoods, A, B, C, and D. And we have a random sample of 650 residents of the city. We take it and we find their occupation, and the occupation can be white collar, blue collar, or no collar at all. So these are their occupations here. So I'll just note this with a blue. This is their occupation. And these are the 4 neighborhoods that they belong to, and we have a random sample of 650 residents. So out of this 1 million, we just took 650 individuals randomly, and the null hypothesis is each person's neighborhood of residence is independent of each person's occupational classification. What that's saying is it really doesn't matter what occupation, if they don't have a collar at all, if they're blue collar, if they're white collar. It really doesn't matter what neighborhoods they choose, whether it's A, B, C, or D. and we have this contingency table, so we're going to do of categorical data. So we're going to do a chi-square test to find out if that's the case. So for instance, let's look at neighborhood A, and we're going to find that, okay, well, in neighborhood A, we have about 150 people here in the neighborhood A. Oh, sorry. $150 people to estimate what proportion of the whole 1 million people live in neighborhood A. So what we find is that, okay, well, we find like we're going to focus on A essentially, and we're going to find that there are 150 people in A, and we're going to focus in particular on the white collar. So we're going to look here at white collar, and we're going to say, okay, well, if we looked across all the white collar people, what would we find? Well, we would find that out of 650 people, 349 out of 650 people in this whole population are white collar. So that's a probability of being white collar in this population. So that's what we would find. So then by the assumption of independence, like if there's really no impact of being being white collar than of living in neighborhood A, then we should expect the number of people in neighborhood A would be the total number of people in our A sample. So number of people in A. So actually, let me write that in red. Number of people in A. And then we, sorry about that, my handwriting is not so neat. So we would want the number of people in A, and then we would want to multiply that by the probability of being a white collar worker. So the frequency of us finding white collar workers. That is what we would ultimately do the multiplication. So that's what this 349 over 650 is representing, and that's what this 150 is representing. So what they're telling us is that if we were to look here, we would find that we would expect to find about 150 people in A, which is what this is, multiplied by the probability of us having a white collar worker. So out of all these 650 people, we have 349 who are white collars, 151 who are blue collars, and 150 who are no collars. So 349 out of them will be white collars. That's what we would expect. So if we multiply them together, we would expect about 80.54 individuals to be living in population A, if they're white collar. And that's what we would expect. And we would also expect, so that's what we would expect this value to be here. This is the expected value. Here, that's the expected value. So we can take that value and plug that in here. And then what we actually observed instead is that there are 90 people. So this is 90 people that we actually found in population A who are white collar. So we would take 90, we would subtract that from our expected values, and then that's how we get 1.11. And it's important, guys, to remember that these are all of our expected value. This is all our observed. This is what we have observed. These are all the observed that we have in our, we have observed here. That's what these values all represent, what we have observed. And these are our expected values are here. So if you kind of wonder, okay, well, what about, we have 1.11, and we're going to do the same thing for all the other possibilities for occupations. And yeah, this observed is representing the whole table. So again, this whole table is the observed, what we actually find in our population. So we'll do this for the rest of the occupations for A, and then also for B, C, and D. So for instance, for B, we also find that B and also D also have 150 people. So they would also have the same expected value of 80.54 for B and for D. We would expect that given the number of people in each group and this frequency of being white collar. So for instance, for A being blue collar, we would expect it to be 30 times no, not 30, 150 times 151 over 650. So if we were trying to look at basically the number of blue collar workers, so if we were looking at blue collar in A, for instance, if we were looking at this, instead of it being 30, we would look at blue collar in A would be 150 times 151 over 650, where this 150 is this, coming from the number of people in A, and then this 151 over 650 is coming from here, the total number of blue collar workers. So similarly, we just calculate all of those squared differences, and then we add them up together, and we get our chi-squared statistic, which here is just is basically in the end we've gotten x squared. The chi-squared value is 24.6, with the degrees of freedom here would be equal to 6, because we have four columns, which is 4 minus 1, multiplied by 3 rows, so 3 minus 1 times 4 minus 1 is 6. So this statistic is probably really large according to that of a chi-squared distribution, so we reject the null hypothesis of independence. If we have a chi-squared value that is 24.6 with 6 degrees of freedom. So we say that there is some association between the person's occupational classification and their person's neighborhood of residence. So what they're occupied with, their occupation and their neighborhood, there is some association between them. So that is statistically significant. So similarly, for our situation, when we're trying to figure out if there's an association between this SNP, this location, and the genotype that we have observed, the first step we're going to do, the number one step, is we're going to first find these total values. So we're going to sum across the disease and across the control, and across these genotypes, and just find the total distribution of individuals in these groups. So again, this is the outcome, the phenotype, and the top is the genotype at this location. So is it a C and a C from each parent, or a G and a G from each parent, or a C from one and a G from the other? Like, does that have any kind of impact on what we observe? And we find that we have about 269 case and or control individuals. So total number of individuals in this study is 269. So again, we're just summing across. The rows for 126 is the sum of all of these, and 143 is the sum of all of this row, and the column total, we just sum 28 plus 95 to get 123, and then we take these two and then these two to get this value. Then what we want to do is we want to then compute the probability of each class, like the case and the control. So the case we typically refer to as the disease, and then the control is the other. So based on this information of the totals, I find that the probability of the disease, so usually it's a mapping from genotype to phenotype in a way. So usually the way that it goes is that we have, use another color, that we have like X is equal to genotype, and then our Y equals our phenotype, like what we observe, like the disease status, like if we have diabetes or not, and we're just trying to map from one to the other. So that's what we're trying to do here. So if we have 126 out of 269 individuals with the disease, then we predict that of having the disease, the probability of having the disease, according to us, is 126 out of 269. So it's similar to the white collar and blue collar, no collar situation, where we just find the probability that we would even observe the disease in the population. So it seems almost rough, almost really close, right? Like 126 out of 269, almost like half in some ways have the disease. And then for the control, what we're finding is 143 out of 269 individuals do not have the disease. That is a probability of each outcome. So that's why we take 126 over 269, and then 143 out of 269. That's a probability of these disease outcomes and control outcomes. And these are the two different classes. And again, this is a contingency table. Then what we want to do is compute the probability of each genotype. So what we find is that, okay, well, we have 123 individuals who are CC. That's why we took the totals. 73 individuals who are CG, and 73 individuals who are also GG. And again, the total was 269. So then we can just take 123 divided by 269. So we can just take these two values together, and then that's how we get this, for instance, this value of this value of 123 over 269. So we do that for the other. We take 73 and then we divide that by 269, and that's how we get the probability of genotype CG. And then we can find the probability of GG as well, because it's 73 over 269 as well. So that's how we find the probability of each genotype. And again, what we also will find is that these are all the metrics that we've gathered: the probability of the disease, the probability of type 1 diabetes here, and then the probability of not having the disease being in the control, the probability of each of the genotypes of interest here in the population. We find that GG and CC are the same, CG are the same, so GG and CG are the same, and the total population count. So these are all the metrics we've gathered in step 4. So then what we're going to do is we're going to find the expected counts. So the expected number of diseases with a certain genotype, CC, CG, or GG. So we're going to use that information on the number of people that we have found in the disease group, number of people in the control group, number of people in our population with each of those genotypes. And we're going to try to figure out how many diseases do we expect for a certain genotype, and how many controls do we expect for a certain genotype. It's similar to what the white collar and occupation, white collar occupation and the neighborhood examples, and the blue collar and the occupation and the neighborhood as well, where we're just trying to figure out, okay, what do we expect the distribution to be given that so many people have these job breakdowns in these 4 neighborhoods, and that we have also observed these number of individuals in each neighborhood. Like, what do we expect the total counts to be if there's no association? Like, what would those typically be expected to be? So here we're going to look at the probability that we have found individuals with CC, CG, and GG genotypes, and the probabilities of us observing the disease and of not observing a disease. And we're going to basically use those to find our expected number of individuals with, with disease who have like CC, genotype, and those who have the disease who have CG, and those who have the disease who have GG. And similarly, we're going to look for the controls and what is expected number of people with CC that we expect to have, with CG that we expect to have, and with GG that we expect to have. If there is independence, if there is truly no association, if the null hypothesis is indeed true, what do we expect? So that's what we do to calculate the expected counts. So to find the expected number of diseases, like, the individuals who have type 1 diabetes who have a certain genotype, what we would do is we will take the probability that we have an individual has a disease in the population, and then a probability of finding that genotype, and then just the number of individuals in the whole population. So there are multiple ways of breaking this down in a way, but this is what this formula represents. Essentially, this is like the key formula that you can use, like having the disease, having that genotype, what are each of those probabilities, and then just a number of people that we observe. And that tells us the total number of counts, the number of individuals with that genotype that we should expect who have the disease. And then similarly, just for the controls, the only difference is that instead of using the probability of having the disease, we just use the probability of not having the disease, of being the control. So these 3 pieces of information is what we had gathered in our metrics in step 4. So we use these. So Sandy says it's just science, right? So I just expanded it out for you guys, and it really depends. Some like SNPs have multiple alleles, so they can have maybe even more than two, though two is typically what we observe for the most part for SNPs. But what we can do is find the expected number of diseases with genotype CC. This is what we would observe. You know, I'm just plugging in the genotypes. So as we go over here, the only difference is that I'm just changing the genotype, you know, based on what we're looking for. So expected number of people who have a disease, who have this genotype of CG, would be the probability of having the disease times the probability of having that genotype CG times the number of individuals in the whole population. And similarly, if you had the genotype GG instead, the only difference is instead I'm taking the probability of us finding somebody who has the genotype GG. And the real difference again between diseases and controls is that I'm just using the probability of us observing the controls in the population. So for very rare diseases, like they have a very low count, then I would expect that these values should be much lower than these corresponding values. And again, just remember that the disease and the controls, they should add up to 1. So, you know, that these values should add up to 1. So similarly, what we can do is we can find the expected you know, number of phenotypes for control people as well. Like what are the expected number for each of these phenotypes that for the controls? So we're going to basically focus on these control individuals and see what do we really expect? Like what is this breakdown that we are expecting? So again, we're focused on these values here, on these control individuals, and on finding, again, what are these breakdowns that we expect. Like, you know, we got 95 individuals who are control, who don't have type 1 diabetes, who have CC, and then 31 CG, who are in control, and then 17 GG as well, who don't have, you know, the type 1 diabetes. But what did we technically expect? You know, for CC, for instance, we have 95 people, but what did we typically expect? You know, we had 123 total individuals with CC, and what we also are observing is that 143 out of 269 individuals do not have the type 1 diabetes. So what do we really expect? So that's what we're trying to find here. So for instance, expected number of controls with genotype CC. So we have the probability of, you know, being a control, which is 143 out of 269 here, 143 over 269. Then the probability of having the genotype CC, which is going to be equal to 123 divided by 269. So both of these are always going to be out of the total number of individuals. And then you multiply it by the total population, which again is 269 individuals in this sample. So that's the total population of the sample. So then what we'll find is that when we simplify this out, we have 65.38622. So that's what we actually would have expected for in here, for instance. So instead of getting 95 individuals based on these results, what we would expect is to have about 65. This value here is what we would technically expect here. So we would expect like 65.39 almost individuals to be in this control for CC. So that's what we expect. And similarly, we also do that for CG as well, and we find like 143 again out of 269. So you're going to notice that these front parts remain the same. So by and large, typically, like when we're looking at the controls, that everything here, these are all the same. The only difference is just the probability of that genotype. So again, like please don't feel like you have to memorize it. Just try to understand the concepts in general. Like these are all the same. The key difference is just this value here. It's just what this is. It's just this genotype. This genotype is different. You know, that's red. This is purple. And then this is blue. This is what's different. And above up here, this is, different. This is what really differs, this probability of each genotype. And we get the probability of each genotype by looking over here at this breakdown: 123 over 269, 73 over 269, and 73 again over 269. So that's what we really pay attention to. That's what really matters in the end. So here we have 38. So this value here would represent what this value here would be, what we would have in place here. So we actually would expect higher than we are having. So we would expect to have 38. So we're having actually lower a number of controls in this group if there was no association between control and CG, for instance, in the genotype. So that could be some flags as well. And this is also a big difference. Like, we actually expect a lot fewer controls of 65.39, and we actually instead have 95 controls with CC. So that's a lot higher than we would expect, given the probability, the prevalence of CC in the population is 123 out of 269, and the amount of controls, which is 143 out of 269. So based on those results, we then just fill out the rest of this table to get our expected counts. And you will see that basically they all should add up. Like a key check should be that if you were to add up all of the rows for each of these across, these should add up to 126. So is that check met? That's what you have to check. Is that met indeed? Like, do these all add up to this? That's your first question. And these similarly, these should also add up going across to 143. And then if you also go down, you should also make sure that 57.61338, this is equal to 123. These should all kind of add up, and by and large they will, based on the formulas that you use. So it's like if I had so he's happy, the star is happy. So based on the information that we have as well, what we will see is that the key idea when we're finding the expected counts is that, hey, we have so many individuals who are in the disease. You have 126 individuals who are diseased, 143 individuals who are control, and 269 individuals overall. And then we have this GG information, CG information, and CC information. These are the breakdowns. So imagine if we just focus on this information here. So if we just focus on this information, on this L, we have just this information. We're going to try to figure out how we distribute it in such a way that we get the counts that we expect. And that's how, like, if there was no association, if the null hypothesis is true and there's no association between this genotype of CC, CG, or GG and the phenotype, disease or control, if there's truly no association, having one genotype does not make you likelier or less likely to have the disease. There's no sort of like observation or trend that we can say that this SNP could be, like, this location on the chromosome could be important. There's no such association of the null hypothesis, the status quo is truly the case, then what would be the expected breakdown, like using all of this data? So if you have all these raw ingredients, you know that there were 126 diseased, 143 control, 123 individuals who have CC, 73 with CG, 73 with GG instead. So how would you arrange all of that information in such a way, like what would be expected if there's no association? Can you distribute it so that the disease adds up here to 126, that like for CG adds up here to the total people adds up to 73 in a way? So it's sort of like telling what do you expect to see, and then we're going to compare this expectation with what we actually find. Like, what did we actually find here instead of those values? What did we actually find in our population? Before we talked that this is 95, but it should be in the 60s according to what we would expect, and then this should be like around 38 from what we expect. So these are the actual observed counts, and then we have our expected. So these parts remain the same. The total counts always remain the same. As we look here, these remain the same here, 123, 73, 73. These remain the same. but it's just these values here that we are like varying based on what if our expectations. These values are what we're changing. You know, so these vary between expectation and observed. Observed is the truth. So that's sort of what how that works. But these are the actual counts, and we have the corresponding set of expected counts. So now what we're going to do is we have these observed counts. You know, this is what we actually find. This is the truth. And this is the expected counts. So now we might be set up for, you know, like some disappointments that, hey, we use the same information in both. You know, we have, again, just to point it out, we have the same set of information here and here as well. This is the same information that we are using here as well. And it's just the difference is, again, just in these breakdowns and in these breakdowns. So you might notice that, hey, okay, this should be, we expect this to be 57, but instead it's 28. So we would actually expect a lot more diseased people than we observed. So we only observed 28 diseased people with CC, but based on the number of CC individuals, 123 out of 269, a huge majority of CC out of all these other genomes, genotypes here, that we would expect like a lot more, like 57, you know, 57 out of the 126 individuals should have the disease, but instead we're just finding 28 out of the 126 have it. So it's sort of like a comparison, you know, like, and these are mapping to each other where we're sort of comparing for each number. What are we actually finding and what did we expect to find? And we're sort of like comparing between these two. We're like, how far off, you know, this is a difference for expected and what is the actual. So this is a lot lower than we expected. It's almost around half of what we expected. So given, you know, that there are 123 out of 269 individuals with CC and that 126 individuals out of 269 people have the disease, we would expect, if there was no connection based on independence, that falling in this genotype and having the disease, if they're truly independent, this is expected as if null is true, what we expect. So this is under the null. Hypothesis. So this is what we would expect. So this is an HO. Under the null, if the null is true under the null hypothesis, then this is a lot lower. So this might mean, and we see here that we observe that this is 95, but we actually thought it should be much lower, of 65. So what's going on exactly? Like, why is it that CC is having such lower counts for disease and higher counts for not having the disease than we expect? And conversely, we actually expect for CG that we would expect 34 individuals to have the disease, but we get even more, 42. So that's higher. And we expected 31 individuals out of the CG, out of the 73, to not have the disease, but we're finding 38.8 actually have it. You know, like 31 do not have the, sorry, we expect that 31 individuals in CG would not have it, but that's what we have observed, but we had expected 38 individuals would have had it instead. So instead of having 38, which is what we expected, we actually only have 31 individuals in CG who don't have type 1 diabetes, and we have a lot more of 42 instead of what we expected of 34 who have the disease. So this is not good. And this is even worse because it seems like having both GGs could be something, because we have 56 individuals who have GG and have type 1 diabetes, which is so much higher than the 34 we expect. So we have expected the same amount for both of these because, you know, 73 and the same overall, you know, total number there. But this is, the difference is far off with having 2 GGs. And in fact, we only expect around like, we expect like 38 individuals again to not have type 1 diabetes to be the control, but we only have 17 that's even smaller. So it seems that as we're going from C to CG to GG, that we're definitely having a lot more, disease risk. The disease risk is also going up. And the difference between the expected and is changing directions, where before this is really good, it seems very protective against our expectations. It's less than what we expected, but it's going up, and the difference is in the positive direction. So again, these are the differences in the squared differences is what we're going to look at, where we're going to take these observed values, like I'll take this 28 and I'll take the expected of 57 and I'll subtract them and I'll square them. And that sort of prevents, like, in either direction, whichever one is greater, the square usually just makes the net result positive. So that's what I do. Like I take 28, which is the observed counts, minus the expected counts, and I square them. And I do that for each of them. So I line both tables up together, and then I subtract like the differences between the corresponding cells, and I square them up together. These counts, like, you can ignore these technically, like I just kept them from part of the original table, but in reality you can just kind of ignore, just ignore these, please. Just ignore that for now. These are just, these values are just the squared values. So yeah, this is step 6, so we're getting through it. And then after we find those fractions of our square differences, which is what this numerator is, so this numerator is what, this numerator here, observed minus expected counts. This is what the square differences is. And then we just divide everything by the expected counts. That's a new thing that we're doing. So we're just dividing everything by the expected counts, and that's what we are getting. So again, they line up together. So maybe a cleaner way of saying that is just showing that that these line up together. Observed minus expected count squared. It's just the same thing as the square difference. And that's what we're doing here. We're just dividing the answers from the previous cell, 876, for instance, by the expected counts, and we're just getting these values here. And that's what the fraction is. And this might look similar to the previous example I was talking with the occupation and the population neighborhood you're living in as well. We had all these values here. And what you're going to notice is in the next step, we're going to focus on these values and on adding up these values. But again, this is just a cleaner version of the same. I just sort of got rid of the math and I showed you what these values are after we have done the calculations of observed minus expected count squared, the square difference divided by the expected counts. This is what we have. And in the end, what we're going to do is we're going to just focus on these values here and on summing them up together. So yeah, that's what we end up doing. We get our chi-squared value by taking our observed counts minus expected counts squared minus expected counts. That is what this whole summation is basically mapping everything to this. We just sum up all these values. So that's where this 15.22133 comes here, plus 1.78235 plus 13. Plus this other 13 here, plus this 1.570462, plus this 12. We sum it all up together and we get, yay, we get our chi-squared value of 58.14697. So that is our chi-squared statistic. Yay, so we have that. So how do we find the degrees of freedom? So the key formula, the DF, that is associated with this chi-squared statistic, so this is called chi-squared. So we look at the number of rows, which here we have, for our number of rows, we have 2 rows. And then we look at the number of columns that we have, which are 3 columns of data. And this works out either way because we're just multiplying them together. We're subtracting 1 from each and we're multiplying them together. So number of rows minus 1 is 2 rows minus 1, which is 2 minus 1, which is 1. And number of columns minus 1 is 3 minus 1, which would be 2. So different from the occupation and neighborhood example, here we only have a degree of freedom of 2 in the end. So 2 minus 1 is 1 times 3 minus 1, which is 2. So 1 times 2 is 2. So 2 degrees of freedom. So that's what we get. So we have a chi-squared statistic from this calculation with 2 degrees of freedom. So again, once we found these observed minus expected count squared divided by expected counts, we just add them up together, and then we get our chi-squared statistic with a certain number of degrees of freedom, which are 2, which we get again by looking at number of columns minus 1 times the number of rows minus 1. So yay! Then the last step, step 9, we're going to just find the p-value for the chi-squared with that is 58.14697, which we found before, that has 2 degrees of freedom. And there are many different tools you can use. You can even look at this chi-squared calculator where you can enter in the original table in. It's going to give you like the frequency as well, breakdown as as well, and it's also going to give you the chi-squared statistic from this table. So you enter it in here, it gives you the chi-squared statistic and the p-value, and the result is statistically significant at a p-value less than 0.05. So you mentioned your threshold. For us, our p-value threshold that we are using is p-value of 5 is significant at 0.05. That's our threshold. So it really depends on your application here. And then if you want more precise, because here it's too small, so it just says it's really, really, really small. But so it's statistically significant. But you can also use software packages like RStudio, for instance, and then this is how you would call it on the console. You would say PCHI squared. You would put in your chi-squared statistic here, this 58 value here. You just put it in here. You put your degrees of freedom in here, and then you have your answer here. So you have 2 degrees of freedom. If you have this value, you just put this value here. And then for your degrees of freedom, you would do this, and that's what happens here. So this is how you would find that, okay, so instead of this being really, really small, it's actually 2.36 times 10 to the negative 13. So again, in regular notation, the way that you would interpret this is that it is 2, let me actually write it like this. Times 10 to the negative 13. Oh, eeks. That's what this p-value is. So the our p-value and what our actual p-value is this. This is what it is, this value right here. That is it, and this is our p-value threshold, and our p-value is definitely lesser than this value of our threshold. So it's actually much smaller than that. So all in all, what we found is that a chi-squared statistic is 58.14697 with 2 degrees of freedom. Again, so it's 2 degrees of freedom. Associated p-value is 2.36 times 10 to the negative 3, which is much less than 0.05. So since the associated p-value is less than 0.05, we therefore had evidence to reject the null hypothesis and accept the alternative hypothesis instead. So the null hypothesis, guys, again, is what we refer to as what this is typically the null hypothesis is our H0, and the alternative hypothesis instead is our HA. So we can conclude that there is statistically significant evidence that there is an association between the genotype at this SNP. So, and basically if it is, if we have observed, so if we have observed like, C, if it's a CC or a CG or a GG, so either of these, that there is an association between the genotype of the SNP and the type 1 diabetes disease status within population A. So we need to do more tests to also confirm our initial thought that maybe having Cs is more protective, at least both C alleles. And if you have a G, like gene over there, the allele, that kind of gives you a higher risk of having type 1 diabetes. That's what we tended to find, but we need to do more experiments because remember, association does not mean causation. Just because things are associated doesn't mean that one thing causes the next. We have to do more biological experiments and statistical tests to see that, but we can look in this SNP, in this region of the chromosome, more carefully after we've identified it from our genome-wide association studies and in our population. We can look at it more carefully and say, hey, what's going on? I think there's an association now between this SNP and the disease status. Either, having this SNP somehow impacts whether or not, you have this disease is associated with you having a higher chance of type 1 diabetes or a lower chance. It has an association, but we need to do more tests afterwards to confirm that hunch of ours. So again, all the steps here, all the steps of this process. So just going over a review again. So we found the total number of individuals in each grouping. We found the probability. The totals are so important because from that we can find the probability of each class, the case and control, the probability of each genotype as well. We gathered all the collected metrics like probability of having the disease, of probability of being in the control, probability for all of the genotypes we have, and we would extend this analysis based on a SNP that has however many different alleles that it can have. Maybe it has 5 alleles instead of 5 possibilities or 6. It really depends. And then we gather all collected metrics. Like for Alzheimer's, they have the APO gene. It can have E2, which is very protective, E3, which is sort of in between, and E4 is really horrible for Alzheimer's. Like if you have both E4 alleles for the APO gene, you have a really, really high risk of late onset Alzheimer's disease. It almost goes up like 15 times. So you could have multiple different alleles, like even more than just C and G. You could have even like 3 different possibilities, for instance. C, E1, E2, E3, E4, and then there are like these 6 different possibilities, if you take all the combinations. So make sure you take whatever I'm showing you and you put it in context based on however many genotypes you have. The same things will transfer over just for the degrees of freedom. Look at the number of columns will change, and you just have to subtract 1 and then multiply it by the disease and the control. So if you have 6 minus 1 is 5, times 2 is 10, we would have 10 degrees of freedom if we had like 6 total genotypes here, and those would correspond to like 3 possible alleles, because all possible combinations of E2, E3, E4 for the APO gene, for instance. Then it's actually a huge risk factor for Alzheimer's. Then we gather all of these collected metrics as well, and then we calculate the expected counts using these metrics. Then we find the square differences for each cell, and then we find the fraction of square difference over expected counts to get our final chi-squared statistic, and then we can find the degrees of freedom as well by number of rows minus 1 times the number of columns minus 1, and then we find the associated p-value for that chi-squared statistic with those degrees of freedom, and then we can determine the statistical significance of the results. So basically, if our p-value is less than 0.05, we reject the null. If our threshold is 0.05, please reject null. I hope that this helped, and then accept the alternative that there is an association. So basically, I really hope that this helped you out. So the null, and then we can conclude there is an association between them, because that's what the alternative hypothesis is saying. So all in all, I hope that this can be a really good framework for you guys to use to understand how the chi-squared statistic works. Thank you guys so much, and please ask me if you have any questions at all. My name is Saniya. Please subscribe to my channel if you like these videos and you find them helpful, and please let me know again if you have any questions. Thank you.